Matrix effect. Matrix effects. Many clinical laboratory methods are affected by the sample matrix, a term for physiological or artificial milieu that contain the analyte. Sometimes two different methods that give the same result for patient sample may give different results when analyzing synthetic samples, such as calibrator fluid proficiency testing, sample and quality control. Ideally, these synthetic samples should mimic patient sample, but many times they don't because the matrix has undergone a type of manufacturing process and doesn't resemble a fresh human patient specimen. The manufacturing process stabilizes and extends the analyte life during shipping and storage, but this process changes the matrix of the native human sample. Calibrator, PT, and QC solution differ from patient sample in that they are supplemented with many different substances to generate wide range of analyte concentration. In addition, these samples are often frozen or lyophilized, freeze-dried to remove all liquid to minimize the composition of analyte during storage. The process of freezing or lyophilization followed by thaw and reconstitution with liquid may also change some properties of the solution when the addition of extra substance or freezing or lyophilization alter the solution properties in a way that biases the measured result. The bias is said to result from matrix effect. When the sample matrix, a term for physiological artificial milieu, contain the analytes contribute to the bias of the measured value, the bias set is to result from matrix effect. Calibrator value are assigned for a specific methods and experiments. If calibrators are used with different methods, matrix effect may result in inaccurate calibration. In PT program, different methods may need to be evaluated using method-specific target values, reflecting varied frequencies of the PT material matrix. The results are separated into peer group of participants who use the same method, the same reagent, or the same analyzer. Ensuring accuracy, clinical laboratory testing must meet numerous scientific and regulatory criteria before it's acceptable for medical decision making. The responsibility for meeting these criteria is shared by the manufacturer and the clinical laboratory. A desirable goal for accuracy is that the combined imprecision and bias should not exceed the typical within subject biological variation, the natural biological fluctuation of the analyte within an individual over time. This ensures that any changes greater than typical biological variability in patient test results reflect real changes in the patient condition rather than laboratory variability. The biological variation is taken into account when test setting the error budget or total error allowable. Total error allowable for a laboratory test is the amount of test results may deviate from the true value and still be acceptable. The TEA has two components, the bias from the true value and the imprecision of the test. Hence calculated bias plus 2 multiplied by CD. For cholesterol, the within subject biological variability predicts that 90% of the test value for the individual would fall within plus minus 10.8% of the true value. The total error budget for cholesterol test should therefore be less than 10.8. The National Cholesterol Education Program requires an error budget for cholesterol certification that's less than 9%. Thus, for NCEB certification, a method with CV of 3% could have a bias up to 3% without exceeding the error budget. TEA 3% plus 2 multiplied by 3%, 9%. Manufacturer responsibility. Before test methods are introduced in the clinical laboratory, the manufacturer must ensure that the test performance criteria fall within acceptable TEA target. The method should be optimized to provide suitable, reproducible, reproducibility and bias, and the calibration process should include traceability to ensure that an accurate value is assigned to the test result. In the US, the Clinical Laboratory Improvement Amendment set the target range for total allowable error for many common clinical chemistry tests. In Germany, the guideline for quality assurance and medical laboratory examination of the German Medical Association defined the acceptable limits. Other professional organizations or regular authorities may also set performance targets for many analytes. Manufacturers must ensure that methods are capable of meeting these targets. Example of TEA target for some selected analytes: albumin, chloride, cholesterol, glucose, total protein, triglyceride, alcohol bleed, thiophyllin. Here the CLIA, here the German guidelines. Clinical laboratory responsibilities. Clinical laboratory must demonstrate that they can perform a test and get acceptable results using the method in a day-to-day operation. Two systems are used to ensure that clinical laboratory are performing acceptably. These are quality control testing and proficiency testing. Quality control testing. In quality control testing of the same sample, a control sample is analyzed multiple times, usually daily, over an extended period of time, weeks to months, to ensure that the test gives reproducible results. The QC results are plotted and graphs called Levy-Jennings plot. These plots show results for analysis of QC sample compared to the expected value and acceptable range of value. The target value and the range of values are determined by the laboratory of each QC sample. Levy-Jennings graph showing acceptable reproducible over one month QC result. This is the days and standard deviation. Typically, 2 QC are monitored for every test. One QC sample is termed normal and has a target value that falls within the expected reference interval for the test. The second QC sample is termed abnormal and has a target value that is outside the reference interval in directions that is clinically significant. When appropriate, the abnormal QC sample will have a concentration that is important for clinical decision making. For example, fasting glucose in non-diabetic is expected to be between 7 and 100 mg per deciliter, and the diagnosis of diabetes is made when fasting glucose is above 126 mg per deciliter. 2 QC sample might be chosen at level of 85 mg per deciliter for the normal QC and 145 mg per deciliter for the abnormal QC sample. If the laboratory deals with patients having low glucose hypoglycemia, it may also include a third QC sample at a low value such as 36 mg per deciliter. The purpose of the QC sample, quality control sample, is to monitor the reliability of the test when the daily QC result falls within the range of acceptable value and scattered symmetrically around the target value. The method is said to be in control. When the results fall outside the target window or show a drift away from the target value over time, the QC results indicate a problem. There are a number of rules that are used to assess the QC pattern and determine when to take action. Usually, action is taken when the number of results that fall outside of the 2 standard deviation range exceeds expectation, or when a series of consecutive results over many days fall on one side of the target value or drift away from the target value. When the QC results show a problem, a systemic investigation into the source of the problem is required. The problem may rest with integrity of the QC sample itself. The problem may be reset with the integrity of the QC sample itself, the stability of the reagent for the test method, the calibration curve, or an experiment problem in a component such as life source, the sampling system, the mixing system, or temperature control. The problem could also lie with the operator and be due to procedural error, staff technique. Before a patient sample can be tested and result reported, the source of the problem need to be identified and corrected, and the analytical system brought back in control. Taking prompt action as soon as QC system indicate a potential problem, ensuring that laboratory doesn't report inaccurate test result on patient sample. Proficiency testing. In proficiency testing, also known as external quality assurance, an external organization, professional organization, or governmental agency send set of unknown sample called BT sample or challenges to the laboratory for analysis. These samples must be treated and tested as if they were patient sample, and the result must be submitted to the BT provider for review. The BT provider usually sends 2 to 5 unknown samples for each analyst and sends sets of samples for all analysts several times per year. The BT provider evaluates the result from many different laboratories and grade the performance based on how close each laboratory result are to the target value for each analyst. Acceptable result must fall within the accuracy range established. For the test in the US, many accuracy range are defined by CLIA. If there is no established accuracy range for any analyst, the test result is compared with the result from other laboratories that tested the same samples. A statistical range is established for all laboratory reporting results for that analyst, or sometimes for subset of laboratories using the same method, termed peer groups. An alternate and more demanding approach to BT is accuracy-based grading. The BT sample are assigned target value by using reference material or methods, and laboratories must recover the target value within predetermined acceptable limits to pass the BT survey challenge. In this approach, the acceptance limits are the same for all laboratories and methods as opposed to peer group grading. If a laboratory fails to perform satisfactorily, the laboratory may be prohibited from performing patient tests for the failed analyst until it can identify and correct the problem and successfully analyze a new set of BT samples from the provider. Only when the laboratory meets the performance criteria for accuracy can it report test results for patient samples.